Hello, everybody. Welcome to Science Abounds Science at Home, where we will discover home lab magic. We will be doing experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and even forensic science. Welcome to my channel. Hello, everybody, and welcome back for another edition of Science Abounds Science at Home, where we will discover home lab magic. In today's episode, we're going to look at the anatomy of the eye. We're going to do a dissection of a cow's eye and examine some of the anatomy that forms an eye. And I'm going to point out some features within the eye that are similar to humans and other animals. And there's a couple of parts of the cow's eye which are a little bit different from a human eye, and I'll point those out as well. Now, if you yourself want to follow along and do a dissection of a cow's eye at home, you can buy kits like I have here from Carolina Biological. They're not sponsoring the video, but Carolina Biological is my resource and go-to place for all my experiments, my lab equipment, my chemicals, kits, etc. Carolina Biological is my number one resource. And they provide dissection kits and organs for dissections. And you can do this in the comfort of your own home and follow along with me. In addition to this video, I've created a document which you can obtain via my email. If you email me, I will send you a copy of this handout that supports the video. It has all the video notes, information about the anatomy and how to perform a dissection. And you can obtain this handout for free if you send me an email making a request. Okay, so these the cow's eye that I have here, it comes in a package. It's preserved. Carolina Biological use a very special preserving agent. It's not formaldehyde, and it's less dangerous or toxic. If you formaldehyde preserved organism, formaldehyde itself is toxic, and you really don't want to be breathing in toxic formaldehyde fumes. But the preservation that Carolina Biological do, it's a safe, harmless preservation solution. It reduces the smell. There's very little smell associated with this, and it's perfectly safe to do the dissection at home, which I'm doing in my home lab. So what it does, it comes in a package like this. So I'm just going to open the package up gently without doing any harm to the actual preserved organ. So I'm just going to snip around the package. I'm sure there's an easier way of doing this, but I'm doing it this way. Like so. Get the package out. So I'm wearing PPE. I'm wearing gloves. You don't want to be doing this without gloves. And I'm wearing an apron So you should wear at least gloves, maybe goggles as well. Okay, so here I've got the organ out, and I'm going to put it on my dissecting tray. So I have a dissecting tray and dissecting instruments. You need at least a pair of sharp scissors. You need maybe some forceps, and you definitely need a scalpel, which I have here. You definitely need a scalpel for this dissection. And like I said, again, my dissection kit is from Carolina Biological, and very inexpensive equipment. And they provide everything you need to do a dissection safely and comfortably in your own home. So let's get up closer and let's take a look at the outer surface of the eye, and then we will begin the dissection. Okay, so we got the eye. Okay, so we got the whole eye, and let's just, before we do any dissecting, let's just look at what we have. So we got the eye here. This is the eyeball. We got the cornea here, this transparent part of the eye. And then we've got all this fat attached and muscle, which we really don't need for dissecting. So I'm going to trim some of that away because it's going to just interfere with my dissecting. So I'm just going to remove some of the fat, or trim the fat off. We don't need it. Okay, and here, this portion here, which I don't want to trim off, is part of the optic nerve. And we'll talk about that in a minute. We're gonna dissect it, and we're gonna talk a little bit part of the different parts of the eye. So what I'm gonna do is I'm just gonna pick up the eye and trim some of the fat off, because I really don't want all this fat on the eye. But I'm gonna be careful not to remove the optic nerve. So this is why you need a sharp scalpel and a sharp knife to remove all this fat that comes attached. Okay. So what I'm going to do is I'm going to trim all this fat, and I'm going to come back to you with all the fat trimmed off. Okay, so I've now trimmed the majority of the fat from the eye. It was tougher than I thought it was going to be. You may want an adult to do that for you to protect your young fingers. But as you can see, I've trimmed most of the fat away now, exposing the eye better. You've got the white sclera surrounding the eyeball. We got the optic nerve here. So this white sclera is the white part of the eye. It's tough, fibrous outer layer that surrounds and protects the inner components of the eye. It also serves, as you can imagine, as structural foundation of the eye, and it has a crucial role in maintaining its shape and its integrity. It's made out of collagen fibers and elastin, which give it strength, but also flexibility, and it gives its white appearance. The outer surface of the sclera is covered by a thin layer of tissue called the conjunctiva, which is clear and helps keep the surface moist. So what we're going to do now is we're going to cut the eye in half with a pair of forceps, with a scalpel. So let's do that now. I'm going to push down. It will require a little bit of effort. Oops. To come down, and be careful because there is liquid in the eye. It's the vitreous humor of the eye. So I want to go all the way around to cut the eye in half. Just make sure you watch your fingers while you're doing this. Okay. And I've almost done it. All right. Oh, look at that. So that's what it looks like cut in half. Okay, so we got the two halves of the eye. We got the back of the eye, and then this is the front of the eye. And as you saw, let me cut this in half because I want to focus on one half at a time. Okay, I still got a lot of fat around the eye that I didn't get rid of. But as you saw, there is the liquid in here called the aqueous humor. There's the aqueous humor, which is at the front of the cornea. We'll have a look in a minute. And then the main liquid in the eye is called the vitreous humor. And the vitreous humor is a clear, it's gel-like. It's not, there is some viscosity to it. I want to try and get as much as I can out. And it fills the posterior chamber of the eye, which is the space between the lens. This is the lens. You may be able to see it here, but we'll take a closer look at the lens in a minute. And the retina. It's the fluid between the lens and the retina, and it serves, and it's about 2/3 of the volume of the eye, and plays several crucial roles in maintaining the eye's shape. It supports the retina. It allows light to pass through to the retina. So the vitreous humor is a gelatinous in nature and consists of water and collagen fibers, hyaluronic acid, and small amounts of proteins. And its ultimate goal is to provide structural support to the eye and maintain a gel-like consistency. So let me just try and remove as much of the liquid as I can. All right. I got some of the liquid removed. So I'm just going to collect some of that liquid from the dissecting tray, which I don't really want. So let's look at the back first. As we know, we have the the optic nerve, which is here. This is the optic nerve. Okay. And the optic nerve is one of the 12 cranial nerves and plays a crucial role in the visual system. It is responsible for transmitting visual information from the retina to the brain, allowing us to perceive and interpret what we see. Now, the optic nerve begins at the retina of the eye at a location which is known as the optic disc. And we'll try and have a look at that in a minute. And the optic disc is also known as the blind spot. And we'll come to that if I can show you, if I can see it. And this is where axons of the retina ganglion cells converge to form the optic nerve. So let's go back in. So this here is the retina portion. And this is what we call the tapetum. It's a reflective layer and is only found in certain animals like cats, dogs, and cows. And it's what causes the red eyes, the eyes to reflect at night. And it gives the creature, the animals, better night vision. And that's where we get the term, cat's eyes for on the roads, the reflective discs on the roads. Well, we call them cat's eyes because of the reflectivity. So I'm just going to take that away because I don't want that part. And that's called the tapetum. Humans do not have this layer. Our eyes do not glow red in the dark. They do reflect light when we like taking pictures. Sometimes you get the red eye, but that's not the tapetum. That's purely because of the retina reflecting light back. So here is the retina. Okay, here. And the retina is a very important part of the eye. The retina, it's a thin layer of tissue, and it lines the back of the eye, and it is crucial for vision. It contains very specialized cells that can detect light and convert it into electrical signals that are sent to the brain via the optic nerve. The photoreceptor layer, which is this is the photoreceptor layer, contains the cells, the rods, and the cone cells. And the rod cells are specialized for color vision and light vision. And the rods are crucial for night vision. They detect light intensity. And then the cones, I apologize. The rods are specialized for light only, not color. And they are receptive to low light and help intensify images during night vision. They detect different light intensity but not color. It is the cones. The cones are responsible for color vision and high visual sharpness. They function best in bright light conditions and allow us to see fine details. So I'm going to see if I can remove the retina. I need my tweezers, my forceps, and I'm going to pull away the retina like so. Okay. And I'm going to try it, and if I can, I may not be able to remove the back part of the eye because I want to show you the optic nerve. So where the optic nerve lies at the back of the eye, it's called, there's a blind spot. Okay. Some of the fat. Okay. I'm not getting. Okay. So the blind spot, this is just part of the eye. So you got the blind spot here. You can't really see it. It's where the optic nerve is connected. And the blind spot is, it's also known as the scotoma. And it's an area of the retina where the vision is not detected. This occurs because there are no photoreceptor cells, there's no rods or cones. And in this region, no light that falls on this part of the retina can be perceived. And this spot corresponds to the point where the optic nerve exits the eye and transmits visual information to the brain. We typically don't notice this blind spot because the brain compensates for the missing visual information. So that's the back of the eye and the retina. So let's look at the front of the eye. Let me see if I can trim some more of the fat. I got quite a lot of fat here. So let me tease that away. Okay. So we have the cornea, and I'm going to try and remove the cornea now. And the cornea is the transparent, dome-shaped outer layer of the eye. And it's the primary refractive surface, helping to focus light into the retina. Okay. The cornea is composed of several layers, including the epithelium, which serves as a protective barrier. You have the Bowen's layer, which is just beneath the epithelium, and is a tough, fibrous material. I'm just trying to do this while talking at the same time. It's not easy. Okay. And then underneath the Bowen's layer, you have the stroma, and it's the thickest layer of the cornea made up of collagen fibers and keratinocytes. And the stroma accounts for most of the cornea's thickness and strength. Then you have the descendants membrane, a thin but strong layer made up of collagen fibers and provides structural integrity. And finally, you have the endothelium, which is the innermost layer consisting of a single layer of cells and plays a crucial role in maintaining the cornea's transparency by regulating fluid and electrolyte balance. So I got some of the cornea. And within this section, you have the aqueous humor layer solution or liquid, just like the aqueous humor, and it's a transparent liquid, just like the vitreous humor of the eye. Let me see if I can snip it around. And basically, the aqueous layer is the layer between the cornea and the lens. Okay. Okay. And that is the cornea. All right, so that's the cornea. And like I was saying with the aqueous humor, it's crucial for maintaining the intraocular pressure, which helps to keep the eye's shape intact. And this is also, this pressure, this intraocular pressure is essential for the proper functioning of the cornea and lens. So we've looked at the cornea. Okay, so what we got left is the iris, which is the color portion of the eye here surrounding the eye. And then the opening, the aperture, is obviously, we all know, is the pupil. So the iris is the colored part of the eye that surrounds the pupil, which is the aperture, and controls the size of the pupil in response to light levels and other factors. It contains two muscles, the sphincter pupillae, which constricts the pupil, and the dilator pupillae, which dilates the pupil. The iris also provides protection against excessive light and UV rays, while its color helps with unique identification. And the pupil is formed from the movement of the iris opening and closing. So the pupil is the circular opening of the center of the iris. It controls the amount of light that enters the eye and reaches the retina, and the size of the pupil changes in response to light conditions. And then, so this is the iris here. You have the pupil. And then at the back, you'll see I'm going to prise away the lens. I'm going to try and remove the lens if I can. May have to use some scissors. Let me try the forceps. This is very delicate to do. Okay, I think I got it. There we go. All right. And so this is the iris, the eye lens. Sorry, not the iris. Usually it's transparent, but because it's been preserved and dried up, it's cloudy. So unfortunately, if it was transparent, you could let this dry out and get some of the transparency back, and you can actually probably read some newsprint if you put this on top of some kind of newsprint or typewritten words. You could probably read or see them through the lens. But right now, I can't see that because the lens is too cloudy to see. So that's the lens. The lens it's a transparent and it's flexible. It's biconcave, and it's located behind the iris and pupil. Its main role is critical role, and its the crucial role of the lens is focusing light onto the retina to form clear images. And its ability to change shape allows the eye to focus on objects at varying distances. Um, and this process is known as accommodation. So this is where, when you need glasses, the lens is not, it loses some of its flexibility and cannot focus as well as it should be. And that's why we have to wear glasses or contact lens to correct that focusing. Well, I hope you enjoyed that anatomy dissection video. I plan to do a lot more of these in the future, so please stay tuned for more of these dissecting videos if it's something you enjoyed watching. Please also remember, if you send me an email, I will requesting a handout. I will be happy to send you a copy of my handout for free. Like I said, it goes over the anatomy of the eye and how to dissect the eye as well. Please subscribe to my channel. It helps me keep going, keep making more and more of these videos. And I appreciate all my subscribers who have already subscribed. Thank you. It means a lot to me. Thank you for your support. Please also hit the like button and the share button, and please leave your comments. Please feel free to email me to share your comments, questions. Send me your photos and videos of you doing your own home dissection. I love to share and build up a home science community. Please check out my description, my video description. I have all my resources listed where you can go to and check out on where to buy science equipment and chemicals and kits, everything you need to do science at home. With that, keep on experimenting. Until next time, goodbye.